Hello dear students, we are going to discuss about the Scoliodon respiratory system. I am Dr. P. Rosina George, Assistant Professor in Geology, St. Joseph College for Women, Visakhapatnam. We are going to discuss about the respiratory organs, mechanism of respiration, physiology of respiration, and the questions based on the topic and the references. The learning outcomes are: you will be able to study the different respiratory organs of Scoliodon. You will understand the mechanism of respiration in Scoliodon, and you will learn the physiology of respiration in Scoliodon. In dogfish, the respiration is aquatic. Since the animal resides in water, it breathes by means of gills borne in a series of gill pouches on either lateral side of the pharynx. There are 5 pairs of gill pouches bearing gills, arranged in a series behind the hyoid arch in the lateral walls of the pharynx. Each gill pouch is compressed anteroposteriorly and communicate with the cavity of the pharynx through a large internal branchial aperture and with the exterior through a narrow external branchial aperture, commonly called as gill slit. The branchial lamellae have a rich blood supply, and they have a very thin covering membrane through which blood is exposed to seawater for an exchange of gases. Each gill pouch has 2 sets of gill lamellae, one on its anterior wall and the other on the posterior. Each set of lamellae is a half gill or hemibranch, so that gill pouch has 2 hemibranchs. The gill pouches are separated from each other by fibromuscular partition called the interbranchial septa. They should more correctly be called intrabranchial septa because each lies between 2 successive gill pouches. The inner part of each interbranchial septum has a supporting cartilaginous visceral arch with slender gill rays. Visceral arches also give out rigid comb-like gill rakers which project inwards to protect the internal branchial apertures from entering the food. Each visceral arch supports the posterior branchial lamellae or hemibranch of posterior gill pouch and the anterior branchial lamellae of the anterior gill clefts. These 2 hemibranchs or demibranchs with their interbranchial septum and the visceral arch constitute a complete gill or holobranch. The posterior hemibranch of a septum has longer lamellae than the anterior one. Thus, a gill pouch has 2 demibranchs belonging to 2 adjacent gills. In dogfish, the hyoid arch bears only a hemibranch on its posterior surface. The first 4 branchial arches has holobranchs and the 5th branchial arch is without any gill. Thus, it has 9 hemibranchs. Between the mandibular and hyoid arches or the first internal gill slit on either lateral side of pharynx is a spiracle. In most elasmobranchs, the spiracle bears minute branchial lamellae forming a false gill or pseudobranch and opens to the exterior by an external branchial aperture. It is supplied with arterial blood and plays no part in respiration. Let's discuss the mechanism of respiration in Scoliodon. Mechanism of respiration takes place in 2 types. One is inspiration and expiration. In inspiration, the floor of buccopharyngeal cavity is depressed by the contraction of hypobranchial muscles, due to which the visceral arches expand the wall of the pharynx, so that seawater containing dissolved oxygen rushes in through the opened mouth. Entry of water into external branchial aperture is prevented by an anterior fold of skin on each gill pouch. In expiration, the mouth becomes closed by the action of adductor muscle. Now the buccopharyngeal floor is raised and the contractions of the wall of the pharynx force the water into internal branchial apertures. The esophagus being closed and then into gill clefts, where it washes the branchial lamellae and goes out of the external branchial apertures. The respiratory movements are caused by pharyngeal muscles, which are innervated by 5th, 7th, 9th, and 10th cranial nerves and the hypoglossal spinal nerve. Let's discuss the physiology of respiration in Scoliodon. In the branchial lamellae, the blood flows from the tip towards the base, that is in a direction opposite to that of the water current, so that the blood just before leaving the lamellae meets the highest concentration of oxygen and the lowest of carbon dioxide. Thus, an efficient exchange of oxygen and carbon dioxide takes place between the blood and the seawater. Fresh seawater entering the gill pouches with the respiratory current contains oxygen dissolved in it. This water is separated from the blood contained within the capillaries of the gill lamellae merely by the thin and permeable membranous walls of the capillaries. The oxygen of the water passes by endosmosis through the thin capillary walls into the blood, and at the same time the carbon dioxide of the blood passes into the water by a process of exosmosis. The oxygen is conveyed by the blood to all the parts of the body, while carbon dioxide brought to the gills in the venous blood is eliminated by the water of the outgoing respiratory current. As the blood makes a complete circuit in the capillaries of the gills in a very short time, it is evident that exchange of gases also takes place very quickly. Now take a break and answer these questions. How many pairs of gill slits are present in Scoliodon? 3, 5, 6 or 7? A complete gill is called hemibranch, holobranch, abranch or lamelliform. Answers are 5 pairs of gill slits are present in Scoliodon and a complete gill is called holobranch. To summarize this topic, the respiratory system of Scoliodon is intricately designed to meet the oxygen demands of this active shark, utilizing specialized structures and mechanisms to maximize oxygen uptake from its aquatic environment. These are the references for this topic. Thank you.